Good evening, everyone, and welcome to the 11th Annual FNIH Awards Ceremony. We work hard to recognize the creative research partnerships that are the hallmark of great team science. But before we really begin our program, I do need to recognize the tragic events that are unfolding in the world around us. These really create kind of a somber background to our meeting. And I know many of your family and friends are affected directly or indirectly by the events in the world, but all of us share in the hope that a peaceful resolution could somehow be found. Thank you. I do thank you for taking time to celebrate something that is life-affirming and reflects the best of humankind, a celebration of some of the finest achievements in biomedical science and progress in healing people and helping them live much healthier lives. So I want you to enjoy your salad course while I'm talking. I'm just going to extend a welcome to some of our special guests. I'm really incredibly grateful to all the dear friends and colleagues who are here tonight, but also our benefactors and our partners from a whole variety of sectors who contribute to the team science that does so much to help patients. I also particularly acknowledge the members of the NIH and the Institute directors and their teams who are here. FNIH exists to support your work, and we will pledge to continue to do that to the best of our ability for as long as we can. I also welcome everyone who's joining us virtually, and especially the FNIH team, many of whom are in the office having a watch party, hopefully enjoying pizza. So I am really honored to share the podium tonight with two individuals who represent our close partners in other parts of government. The acting director of the NIH, Dr. Larry Tabak, and Dr. Robert Califf, who's the commissioner of the Food and Drug Administration. Dr. Monica Bertagnolli, who had a very long day today in her Senate confirmation hearing and did a brilliant job for all of us. So thank you, Monica. You're going to hear more from our awardees and the recipients of the Lurie Prize, the Trailblazer Prize, and the Charles Sanders Partnership Awards a little bit later. But I do want to especially thank Anne Lurie, Drs. John and Elaine Gallen, and Charlie Sanders, who could not be here this evening, for making this evening and these awards possible. Their continued support and commitment to supporting the science stars that we honor today is really a precious gift. I also especially welcome and thank my friend and our event chair, Fred Siegel, and his guests. Fred exemplifies what it means to truly commit to a noble cause, and we are blessed by his leadership, his hard work, and by his inspiring ideas about how we can make this whole event more fun, bigger, and more powerful in raising the resources that we need to be able to jumpstart some of the science that we're trying to accomplish. Thank you, Fred. Now, some of you may know that we've launched a new strategy at the FNIH in support of the mission of the NIH, and that is the Building Bridges to Breakthrough strategy. We chose the framework very carefully because we want to signal the role of NIH as a key bridge between government scientists and those other sectors of the health ecosystem. So we kind of had a chance earlier this evening to appreciate the venue of the REACH, this part of the Kennedy Center, the water, the sky, and hopefully the bridges that cross the Potomac River. We think this was a great metaphor for our strategy, the Building Bridges to Breakthrough strategy. And if you go online and look at the video called The REACH, which is the story about this venue, you will hear John F. Kennedy's granddaughter, Rose Kennedy Schlossberg, state, "My grandfather believed in building bridges." And then later in the video, the Kennedy Senator President Deborah Rutter says, "The REACH was created to build bridges, cross boundaries, unite, and inspire." Now, she was talking about the arts, but I would contend that science is a form of art, one to be cherished, promoted, and protected at all costs. FNIH's work in building bridges between the extraordinary NIH scientists and their counterparts in life sciences organizations, in patient organizations, in academia, and many other partner organizations. This is exactly what we were authorized by Congress to conduct more than 25 years ago, and we hope to be able to continue and expand and accelerate that trajectory. So let me really, again, thank the NIH Institute and center directors and their colleagues. They are really powerful instigators of the unprecedented progress in science that has been accomplished so that we're really able to imagine things today that we couldn't have even thought of 10 years ago. So thank you for your service. And I would like to give the NIH leadership a round of applause. Thank you. So with that, let me introduce our video that I think tells the story of bridges to breakthroughs far better than I can tell it from this podium.
My daughter Taylor, when she was diagnosed at the age of 7 with CLN1 Batten disease, the geneticist said, "There is nothing you can do." I knew that I had a new goal in life. If I couldn't help my own child, there had to be an answer for families and children who would follow my own.
Science has the power to cure, but no single organization can do that alone. At FNIH, we build bridges to breakthroughs by forging teams of the very best scientists at NIH, in academia, and across the biopharmaceutical ecosystem. Through the public-private partnerships, we accelerate the creation of new diagnostics, therapies, and potential cures.
FNIH has been charged by Congress to really fill a gap in the ecosystem of innovation.
Before FNIH, it was really difficult to establish collaborative efforts between public and private sectors.
FNIH brings its own special expertise to putting these together in a way that can really be productive.
The FNIH is the consummate convener.
And that means bringing together the best science and technology, outstanding people who use their combined expertise and ambition to accelerate progress for patients.
The more hands you have on deck, the more likely you are to answer these vexing problems.
We bring together the resources, we provide the infrastructure, and really form that bridge to the breakthrough.
At FNIH, we support team science aimed at solving some of the toughest biomedical challenges. Since our inception, we've raised more than $1.5 billion to support the mission of the NIH and have deployed those funds for programs like Accelerating Medicines Partnership and the Biomarkers Consortium. These programs bring treatments and, most importantly, hope to people living with cancer, neurologic disease, rare genetic disorders, and other seemingly incurable health conditions.
With every partnership we foster, we're seeking to accelerate scientific innovation so that we can realize global health equity in our lifetime.
We're working with NIH, the Gates Foundation, and many other partners on some of the grand challenges in global health, like maternal mortality, malaria, tuberculosis, and HIV/AIDS.
The discoveries that happen through FNIH affect the health of everyone in the world.
These breakthroughs wouldn't happen without world-class scientists. That's why we take time to honor the creative investigators who are pushing the boundaries of possibility with awards like the Lurie Prize in Biomedical Science and Trailblazer Prize.
The FNIH has supported nearly 25,000 scientists over the last 25 years with training programs, fellowships, and awards.
Our partners at NIH and the academicians they fund make discoveries in their laboratories. Our partners in life science companies discover and develop medicines for patients. FNIH builds the bridge from the bench to the bedside and reduces the time it takes to bring these promising therapeutics to patients who can't wait.
Taylor lived almost 13 years with her diagnosis. I want mothers that follow me to have more. The FNIH is running a great race on behalf of people like my daughter.
I know of no other organization in the world that could do this the way that FNIH does.
But we can't do this alone. We need your support. So please join us in building bridges to breakthroughs. Patients everywhere are waiting.
Thank you. I feel inspired every time I watch this video, and I really want to thank David Carmel and the communications team and their support efforts to really not just modernize our website, but to really build the stories that we need to tell about the important work that goes on in this organization. So if you want to log into our website at fnih.org, you will see many other examples of stories that I think you'll find equally inspiring. We did have a great year, and I don't have time to talk about all the highlights, but I want to just make a couple of comments. February was incredibly exciting to a whole community of people who care about maternal health because the New England Journal published the A+ initiative, which was led by NIH's Eunice Kennedy Shriver National Institute for Child Health and Human Development. The study showed that a single dose of an antibiotic, azithromycin, given during labor could reduce the risk of maternal sepsis, life-threatening infection, by 35%. This is an amazing intervention for women in the developing world, and when this drug becomes widely available, it is a generic, it's very inexpensive, and this will save lives of many, many mothers around the world. In May, we marked the announcement of the 8 first diseases that were selected for gene therapy in our Bespoke Gene Therapy Consortium. This is an initiative that hopefully will bring hope to many families, but in addition will help us understand what are the good manufacturing practices for creating gene vectors and what are the regulatory pathways that will help achieve approval of these promising therapies, even when we can't do randomized phase 3 controlled clinical studies because there simply aren't enough patients with rare disease to populate the study. In June, we began a project to explore the use of gene therapy that will actually cure sickle cell disease, an approach that hopefully will eventually be helpful to the people in the underserved areas of the world who are most likely to have sickle cell disease. This approach also holds promise, we hope, to be able to do the same someday for HIV. This year, we also welcomed 2 wonderful new members to our board. Jay Bradner, who was the president of the Novartis Institute for Biomedical Research and who is a practicing physician scientist at the Boston Medical Centers, and Jim Weiss, who is the founder and chairman of Real Chemistry, an important firm that has a great deal of engagement from a communications perspective with the life sciences industry. Both Jay and Jim bring a wealth of knowledge and experience to our board, and we welcome them and also thank Steve Paul, our board chair, and all our other wonderful board members for their wise counsel. Their support means everything, and they are very actively engaged in our success. We also have some things to look forward to in 2024. We will be launching yet another Accelerating Medicines partnership. This one is focused on ALS, or Lou Gehrig's disease, which many of you know is a fatal neurodegenerative disease for which there currently exists no cure. And in February, we're going to celebrate the 10th anniversary of the collection of these programs with a 2-day summit here in Washington, D.C. And in May, thanks to a former board member, Joel Marcus, we will convene at the Alexandria Center for Life Sciences in New York City for our first ever Team Science Day, when we bring together our NIH colleagues, our other scientific partners, and other stakeholders to hear about our science and to get updated on the state of the art. We will be including many stakeholders, but also the science media in this event. So I really hope that you get the impression, or I'm building the impression here, that we truly are committed to building bridges, and we aim to be exemplars in supporting team science at its best. For everyone who sponsored this event and the science that underpins it, we thank you. I'm especially grateful to our premier sponsors, Dr. Steve and Jan Paul, and Fred and Donna Siegel, and our visionary sponsors, which include GSK, the Judy and Peter Blum-Kovar Foundation, Dr. Gil Oman and Martha Darling, and the Edmund J. Safra Philanthropic Foundation. And to all our partners, our board members, our NIH leaders, and our guests and benefactors who are investing in science, thank you. My most important heartfelt thank you to my husband and constant companion, David Rose, who joins me tonight. Thank you, David. So now, having endured the preliminaries, Monica, I'd like to invite Larry Tabak to join me at the podium. I have had incredible appreciation for Larry's candid counsel and his mentoring this past year. Serving the acting director of anything is never an easy role, and I would say serving in this capacity at NIH has had to be incredibly challenging. I can't imagine anyone more suited to take on that responsibility during, shall I say, these unprecedented times than Dr. Tabak. So please join me in thanking him and welcoming him to the podium.
Well, thank you very much, and I'm really delighted to be back at the podium as part of this celebratory event. It feels great to be in this mix of individuals and organizations who are so deeply interested in accelerating and advancing health science. Now, as you've heard, I'm a short timer, and that's great. But I do welcome the opportunity to highlight the ongoing partnerships between the NIH and this foundation that has been supporting our initiatives so ably. FNIH has been a steadfast partner in promoting an immense spectrum of initiatives. They have accomplished this through their unparalleled ability to convene impactful public-private partnerships, bringing together an impressive brain trust and the necessary resources to jumpstart and accelerate innovation. The FNIH aligns with us on our many programs and initiatives that celebrate and train the next generation of scientists. And this year, leadership teams at the NIH and the FNIH have been gathering to explore even stronger paths of communication and collaboration. These meetings so far included bringing together the Foundation with the NIH's Fogarty International Center, the Eunice Kennedy Shriver National Institute of Child Health and Human Development, and the National Institute of Mental Health. Together, the teams discussed potential collaborations involving such things as new biomarkers, medical devices, global research and data sharing, and the concept of One Health. We certainly look forward to continuing to pair our knowledge and experience that will build more bridges to a healthier future, and I, for one, I'm very excited about both the 10th anniversary AMP Summit in February and the FNIH's premier Science Day in the spring. Your partners at the NIH will be with you every step of the way to help ensure that both of these events are enormously successful. I'm still going to propose the toast. So I want to propose a toast to my dear colleague, Julie Gerberding, to her talented staff, to this evening's awardees, to those who have supported this event, and to all of you who have come here together tonight to underscore the importance of scientific discovery for the good of all. So please raise your glasses for a toast. Hear, hear. And since we are on the subject of partnerships, I would now like to invite my colleague, Dr. Rob Califf, to the podium. Rob, I want to thank you for your really extraordinary leadership at the FDA and for your unprecedented strong partnerships with the NIH. So thank you all.
Thanks, Larry. It's great to be back for my annual report. And I have to say, it is remarkable to see so many good people in one place, and just to reflect on how important these sorts of partnerships are. But let me just get right down to business on the annual report. I've heard a lot about developing countries. Last year, I probably talked too long about the state of the United States, but I was just in Singapore 2 weeks ago. Their biggest problem is that the average person will live to be 85, and their average teenager today will live to be 100. And they're trying to figure out what to do with that. There I was with my United States government contingency. We're at 76 and going down. We're currently in last place among high-income countries in life expectancy. So as we focus on discovery, and I'm all for it, I want to have new things, say a little bit more about that in a minute. This needs to be the time when we focus on how we implement what we know in a way that leads to better health outcomes for the people that live in this country, because we are headed the wrong direction in a hurry, and I think this is an emergency that we need to wake up about and get really focused upon. Now, the second part of my annual report is about Larry Tabak, who I feel like last year I think I gave like a whole long litany of his many attributes, thinking that he would be gone by now. So I won't repeat them all, other than just to say that, having been part of the HHS family and in my first term as commissioner, Larry has bailed me out a number of times. He knows everything there is to know about the bureaucracy of the federal government and how to optimize its function. And for that, I'm eternally grateful. But I think we all should be grateful for the amazing job that he's done as a stand-in. And but I think a lot of people know how excited I am. And this gets back to my first comment on the state of this country, health-wise. It's time, from the FDA perspective, I feel like this country, and everywhere I go, people say the same thing. We are the world leaders in innovation. We're the world leaders in developing new medical products. The technology we developed is awesome and has a lot to offer people. But as I said, we are at the bottom of the heap when it comes to deploying the technology that we have and the health resources that we could put into play. And I was really thrilled. I heard Monica say it in the hearing, that we're going to focus on the clinical trials arena. From the FDA perspective, that space, after products are approved and they're on the market, we don't know what to do with them right now. And we have no system that delivers systematic information that would inform a patient, a doctor, a nurse practitioner, a pharmacist, a health system administrator, or an insurer about how to choose one over the other. It's almost as if we designed a system that was intended to never answer that question, so we could just leave it to advertising and detailing as a way to get this done. And the result of that, in my opinion, is $4.3 trillion a year with the worst outcomes of any high-income country. Have I made my point? I'll stop at that. Last thing, I do want to celebrate innovation. And it's special for me, as some people know. I'll just say it. I have a younger brother who has pancreatic cancer, and so I was really excited to see that one of the awards this year is for people working in that field. And so I do have a deep sense of the urgency that we all have when we have a critical illness, and we're hoping there's some breakthrough that will make a difference. As I told people the other night, it's a little different. My brother's an orthopedic surgeon. He knows a lot of smart doctors, and he happens to have the head of the NCI and the head of the drugs part of the FDA looking in on his case, so he's getting pretty good support. But without these bridging functions that we're talking about now, we would have so much discovery, new ideas that never make it to the place they need to be. And without a clinical trial system that really works in this country, we're not going to know what to do with those technologies when they get there. So very excited about what the future has to offer. Let's all work together and make it happen. Thank you.
We're going to go ahead and continue with our program at this point. Thank you. So One of the things I learned when I served as the chief patient officer at Merck was the value in making sure that patients were also always the center of everything that we do. So I was so pleased to come to FNIH and realize that the same was true in this organization. Everyone is committed to the same ultimate purpose. The thing that gets them up in the morning is the sense that we need to act with urgency so that the impact of our work can really benefit patients and their families faster. So we are really working hard to be exemplars of patient centricity in all of our work, from the bench to the bedside, so to speak, and beyond. This year we launched a new patient advisory council to bring patients and members of their communities in to advise us. And we also launched a patient ambassador program to ensure that the lives and the lived experiences of our patients were available to us, not only to inform our work, but also to help let others in the world know how important this was to us and how we could connect with them. So tonight, we're honored by the presence of a very special guest. Carlos Lorari, who's one of our first patient ambassadors, has selflessly shared his experiences to help improve our scientific partnerships and ensure that they are exemplars of patient centricity. So, Carlos, thank you for being here for this and for all of the things you do to support the FNIH. Your work really matters. Please join us.
Thank you, Julie. Good evening. My name is Carlos, and I am Steering Committee Co-Chair for the Accelerating Medicines Partnership for Schizophrenia and former board member of NAMI, the National Alliance on Mental Illness. I'm an advanced practice nurse by training, a law student at the University of Michigan, go Blue. That's right. A public administration student at the Harvard Kennedy School, and a member of the first cohort of the FNIH Patient Ambassadors. Most importantly, I'm also an individual living with schizophrenia, and I hope to briefly share my story today. In adolescence, I began noticing changes in my mental health, with thoughts resembling a broken radio and unexplained sadness. Despite excelling in high school and gaining early admissions into medical school, my college years marked a stark decline. I started hearing voices and becoming obsessed with the idea of being the Messiah. By my senior year, I was isolated, neglecting my hygiene, and rambling incoherently. Fortunately, a friend at college I'd known from home called my mom and said, "Carlos's behavior's off. You need to talk to him." My mother arrived on campus the next day, and the moment that changed the course of my life, I had a meeting with my academic advisor and my mom. My advisor said, "Carlos, you're an adult. You have a right to privacy. I don't have to discuss anything that's happened to you on campus with your mother." I said, "With all due respect, Dr. Clark, I have a Cuban mother, so I've never really had a right to privacy." That is a true story. And that moment opened the door for an honest conversation about my mental health. The fog of psychosis subsided for just a moment, and I was able to take my first steps towards recovery. My lived experience has been a challenging journey, no doubt, but my recovery has also taught me about the immense capacity of the human spirit for resiliency, empathy, and connection. It has also compelled me to advocate relentlessly for myself and others living with this condition. And I firmly believe that when striving to find better diagnostics, therapeutics, and cures for patients living with disease, the patients and their families must be at the table. Only through the inclusion of those directly affected can we genuinely alter the course of their lives for the better. Bringing patients into research and policymaking builds trust, builds legitimacy, and fosters the development of outcomes and solutions that matter to patients and their families. As patients, we need to be part of the conversation as it's happening, and I'm proud to say that the FNIH is making significant strides, actually serving as an example for others in biomedical research of end-to-end patient exclusion from the very inception of a project through planning and clinical trials to outcomes. Because of my early diagnosis and my involvement in organizations like the FNIH and NAMI, advocating and working to better understand and treat schizophrenia, I am fortunate to live a full and meaningful life. But others have not been so lucky. I would like to acknowledge the FNIH's Director of Patient Engagement, Tanya Kamphaus, here with us tonight. Thank you. She has been a tremendous force in building and launching this initiative. And I salute all the people in this room, the astonishing endeavors by those receiving awards tonight for their work, and the team at the FNIH for taking the necessary steps to include me and my colleagues in moving medicine into a brighter future. Thank you.
Yeah, thank you, Carlos. Really appreciate all you're doing for us and for making time for this evening. Thank you. So we're moving to the part of the program that you're waiting for to learn a little bit more about our awardees tonight. To introduce the Trailblazer Prize for Clinician Scientists, I invite Dr. John Gallen to the podium. I know you know him as the longstanding director of the NIH Clinical Centers. He and his wife, Elaine, are lifelong champions of the value of clinician scientists and their unique role in the research ecosystem. They instigated the Trailblazer Prize Award to ensure that clinician scientists continue to be recognized and celebrated. Thank you, John and Elaine. Thank you.
Thank you, Julie. I'm John Gallen, and I'm delighted to be here with everyone tonight, both those in the room and those who are online. My wife, Dr. Elaine Gallen, who's sitting at the table, and I are thrilled once again to celebrate the truly game-changing work of this year's recipient of the 6th Annual Trailblazer Prize for Clinician Scientists, Dr. Vinod Balachandran. The more than 5 decades I have spent at the NIH, first at the National Institute of Allergy and Infectious Diseases, and then at the Clinical Center, and my wife's past work supporting clinical investigators at the Doris Duke Foundation and advising philanthropic organizations led us to partner with the FNIH in 2018 to establish the Trailblazer Prize. The Trailblazer Prize recognizes the critical role of early career clinician scientists in translating basic science discoveries to the bedside to ultimately improve human health. By celebrating the outstanding contributions of clinician scientists whose groundbreaking work can transform patient care, we hope to inspire others to follow in their footsteps. An exemplary jury of clinical investigators reviewed the nominations of the Trailblazer Prize submitted from many of the very best academic medical centers in the country. The jury, chaired by Dr. Michael Welch from the University of Iowa, was thrilled to award this year's prize to Dr. Balachandran, who epitomizes the goals of the prize. In addition to tonight's celebration, we are also pleased to announce that the FNIH has arranged for Dr. Balachandran to be recognized for this prize by presenting at next year's National Clinical Research Meeting of the Association of American Physicians, the American Society for Clinical Investigation, and the American Physician Scientists Association. Dr. Balachandran serves as a physician scientist in the Immuno-Oncology Service of the Human Oncology and Pathogenesis Program and as a surgical oncologist at the Memorial Sloan Kettering Cancer Center, where he specializes in treating people with pancreatic cancer. He is also immunology group leader in the David M. Rubenstein Center for Pancreatic Cancer Research at MSK. By tracking and studying the very rare long-term survivors of pancreatic cancer, Dr. Balachandran was able to show these patients have unique tumors enriched in neoantigens, proteins recognized as foreign that arise from cancer mutations, which can trigger immune cells to keep pancreatic cancers in check. With the help of NIH funding, he is now translating these laboratory discoveries into new immunotherapies for pancreatic cancer, including the first clinical trials of personalized mRNA vaccines that target neoantigens from a patient's tumor, stimulating the patient's immune system to attack the cancer. A very exciting step forward. This pioneering work has the potential to transform the treatment of pancreatic cancer, one of the most aggressive and difficult to treat cancers, and to provide proof of concept for future cancer vaccines. In addition to Dr. Balachandran's outstanding research, the jury was delighted that he is a trailblazer who is also an outstanding surgeon. We rarely see active surgeons who find time to conduct this level of clinical investigation. His efforts translating basic science discoveries quickly and safely into treatments for patients with pancreatic cancer demonstrate the innovative research that we celebrate. Dr. Balachandran will now share a few words about the work that he's passionate about. Dr. Balachandran, please come to the podium to share your story.
Thank you, Drs. Gallen, the FNIH, and the Trailblazer Prize Selection Committee for conferring this great honor on our team and shining a really much-needed spotlight on pancreatic cancer. So in 2023, 90% of pancreatic cancer patients die within 5 years of diagnosis. As all current treatments remain ineffective, in just 1 more year, pancreatic cancer will become the second leading cause of cancer death in the United States. We urgently need new ideas. So what was ours? So it turns out that 90% of pancreatic cancer patients do die, but not all do, and a rare 10% survive long-term. Why? Unknown. So for the past 8 years, we've studied these rare survivors with 2 questions in mind. How do they do this, and how can we replicate this? And what these patients have taught us was that each of their immune systems recognized foreign proteins or antigens in their cancer, which was potentially contributing to their exceptional survival. So we set out to isolate these antigens to perhaps teach other patients' immune systems to similarly recognize their cancers, like what may be happening spontaneously in pancreatic cancers, by delivering these antigens as vaccines. So it turned out each antigen in these patients' tumors was different. So each vaccine would have to be custom-made for every single patient. And in 2017, we thought this was feasible with mRNA. And in 2019, we launched the first clinical trial of mRNA vaccines for pancreatic cancer, historically a cancer thought to be vaccine unsuited. Here, in this trial, we performed complex cancer surgery to remove pancreatic cancers at Memorial Sloan Kettering in New York. We shipped the cancers to Germany in 3 days. We custom make an mRNA vaccine for each patient, and we treat patients in 6 weeks. Now, right after we started, of course, the pandemic struck. Hospital shutdowns in New York, supply chain disruptions, which threatened to derail the real-time cross-Atlantic transfer of patient material and drug which was required for this trial. And our partners, a then little-known company called BioNTech, was now distracted trying to save the world from COVID-19. But despite these challenges, we completed this trial a full year ahead of schedule and learned that mRNA can make fast, potent vaccines that can teach the immune system to detect even the most difficult to detect cancers, results that we are now testing in patients around the world. But our inspiration stems from patients, long-term survivors who send their blood to us by FedEx from all over the world, the survivor who gave me permission to perform a house call and draw her blood in her living room so that we can study it in the laboratory, the patients who travel 12 hours to New York every week for 9 weeks to receive their mRNA vaccines. They are the real trailblazers who overcome challenges that for us are really impossible to fathom. So thank you to all the trailblazing patients. You are really our inspiration in our quest for progress against this deadly cancer. And thank you all.
Okay, so we will now bestow the award from the FNIH Foundation for the National Institutes of Health presents the 2023 Trailblazer Prize for Clinician Scientists to Vinod Balachandran, MD, for his pioneering contributions to the field of pancreatic research and treatment. Congratulations. So Elaine joins me in saying thank you, Dr. Balachandran, for your work and extending congratulations for your outstanding contributions. And we look forward to hearing more about you and your exceptional accomplishments in the future.
Welcome back. This year is the 11th year of the Lurie Prize. Recipients are truly superstars and operate at the cutting edge of pioneering science. Just some of the areas of research that have been honored in the past, including the deployment of super resolution microscopy, explaining both the positive and negative attributes of the microbiome, the development of CRISPR, which, as you know, led to our Nobel Prize winner. And last year's recipients, one of whom's here with us tonight, Dr. Andrew Dillon. Andrew, where are you? Thank you, Andrew. Thank you for returning. He and his counterpart conducted separate but complementary studies on regulating aging at the cellular level. So the science is diverse but excellent. And once again, we're honoring two exceptional individuals who have dedicated their research and their lab teams to distinct but equally exceptional areas of work, exploring the function of mitochondria. Anne Lurie has given us a great honor in allowing us to acknowledge these and other recipients of this award year after year. And I have a wonderful announcement to share tonight because Anne has agreed to fund this National Recognized Prize for an additional 10 years. Please join me in thanking her. So let's now turn our attention to our Lurie Prize winners, Drs. Navdeep Sandel and Vamsi Mutha. But before we bring them up here to talk with us, let's watch a video. Can you roll the tape, please?
My name is Vamsi Mutha. I'm a professor of systems biology and medicine at Harvard Medical School, and I'm also an investigator of the Howard Hughes Medical Institute.
I'm Navdeep Chandel. I'm a professor of medicine, biochemistry, and molecular genetics at the Feinberg School of Medicine at Northwestern University. I'm thrilled to receive the Anne Lurie Prize this year with Dr. Mutha.
I feel so honored to be receiving the Anne Lurie Prize. I'm deeply humbled. Dr. Chandel and I both share a passion for mitochondria and its role in human disease.
Biology is rooted in observations. Of course, then after you make an observation, you have to use the logic and physical chemistry principles to figure out the mechanism of why that observation works the way it does.
Mitochondria are semi-autonomous organelles that are found in all of our body's cells that help to extract energy from the food that we eat and convert it into a chemical energy that then powers everything from growth to neurotransmission.
That's why it's popularly known as the powerhouse of the cell. But what we've learned in the last 25 years is a whole new biology of mitochondria.
This is a very, very complex organelle, so it really benefits from a lot of different approaches. And I think we've both had very parallel bodies of work that helped illuminate some basic aspects of the organelle as well as its role in disease. In a series of papers reported in 2016 and 2019, we discovered that if we simply dial down the ambient oxygen, we could now suppress disease in preclinical models of mitochondrial disease. And conversely, if we dialed up oxygen, it was a great way of making those organisms even more sick. So we think we have uncovered an extremely deep and profound interaction between mitochondria and environmental oxygen. We're now trying to harness hypoxia as a medicine for both rare and common forms of mitochondrial dysfunction. Also, for almost 20 years now, we applied new methods such as mass spectrometry, proteomics, and big data computational analysis to try to systematically identify all of the proteins that are made by the nuclear genome that then end up in mitochondria. So by using these methods over many, many years now, we've identified about 1,100 proteins that we call mitocarda. And these 1,100 proteins made by the nuclear genome work with the 13 that come from the mitochondrial DNA. And all of these have to work together to produce properly functioning mitochondria.
This paradigm that I've worked on since the mid-90s now, called mitochondria signaling organelles. Mitochondria release other things other than ATP, which make critical decisions for health. And if those signals are perturbed, then one idea is that that's why you get pathology. Maybe that's why you get neurodegeneration, or you get too much inflammation. And so if we figured out some of those signals, others have figured out other types of signals, then we can figure out how those signals are generated and released and what their targets are, and how those signals then change during a particular pathology. Then let's fix those signals and cure the pathology. So hence I've sort of coined this mitochondria signaling organelles. So in some ways mine is a reductionist approach. His is maybe a systems approach. They're both complementary. And I use some of these systems approaches to figure out where to focus in an unbiased way. And he of course takes his systems when he wants to do mechanism and causality, he goes to the reductionist approach. So I think it's a nice sort of yin and yang.
My real hope is that we can just continue doing this work. We're always driven by just basic biology as well as by disease biology. I would love it if we can change the textbooks of biochemistry and also change the way that we practice clinical medicine.
I'm hoping now that the concept of mitochondria signaling organelles is formally established in my lab and in many other labs. People are discovering these new signals coming from mitochondria beyond ATP, and how those signals change in a given disease. And if we figure out how they change, maybe we can fix them to develop therapies. And that's what we're gonna try to do going forward.
I really want to thank the FNIH for honoring me. This really helps to recognize all of my mentors and all of my lab members. So by honoring this field, you're bringing a lot of attention to the science and medicine of mitochondria.
I'm very thankful to receive the Anne Lurie Prize. You're only as good as the people in your lab, and this is bestowed upon the Chandel Lab collectively.
This is a really exciting area that holds so much potential for all of biomedicine.
To tell you that in preparation for this evening, I had the chance to talk with both of these scientists at length, and then I wouldn't say plagued, I was provided with lots of scientific articles to read, and I just became fascinated with mitochondria. Of course I should be, because I'm an infectious disease clinician, and after all, mitochondria are just little bacteria that found their way inside of the cell. So they do have their origin in an area that I'm familiar with. But you know, when I had a chance to talk with both of these scientists, I realized that they do have a lot in common. Obviously, they're both of Indian descent. Both of them, at earlier stages of their career, were interested in mathematics, a point that I'll come back to in a second. They evolved under the tutelage of some extraordinary mentors, and you both have made reference to the importance of the mentors in the lab environments that you evolved your interests in. And independent of each other, operating in very distinct environments, they still simultaneously have developed this incredible passion for mitochondria. I don't know what you talk about when you have a glass of wine together, but I can guess. So I'm going to start with you, Nav, and just go back in time. You got your BA in mathematics from the University of Chicago. That seems kind of a far away distance from what you're doing right now. How did it happen that you found yourself with a PhD in this area and went on to develop this obsession that you have?
It's pretty simple. It turned out I wasn't going to be a great mathematician. Even though I got a math degree from the University of Chicago, which is a very difficult place to get a math degree, I wasn't going to solve Fermat's last equation or something important like that. I've always told my own trainees and people, one of the beautiful things about biology compared to math is math actually has an intellectual ceiling, and biology is an observational science, right? And I think I really genuinely mean this. I think anyone can make an observation if they really try hard enough and they're dedicated to it. You go in the laboratory and you make an observation. Of course, the key is sometimes someone's observation might yield a breakthrough and others doesn't. And why that is and how people pick which observations to go after is the secret sauce. But unlike math, after a while it starts to look like hieroglyphics or ancient Greek to me. Where in biology, every day someone in the lab makes an observation, and that can excite you and push you into different directions. And sometimes those observations lead to breakthroughs, and sometimes they don't, but that's okay.
Can you say a word about your mentors along this pathway?
Yeah, so I was blessed with 3 mentors, Paul Schumacher, Celeste Simon, and Craig Thompson. They were all very different. Paul is an engineer by background and also has a math, so rooted in logic. Craig Thompson, who ran Memorial Sloan Kettering, that Vinod probably knows well, is a big thinker. Celeste Simon told me the importance of always use genetics to show causality as a way of illuminating biology. And I think for me, this was the perfect combination. I always say I did the shortest postdoc of any living person that I know because I had 3 mentors. Right? Typically, you do a 6-year postdoc. I ended up doing 2 because I had 3 mentors. You could just squeeze them all in 2 years. So I was blessed by having the 3.
So Vamsi, let me talk with you a little bit about your background in mathematics. So you may not know this because it's not necessarily in his bio, but he did win the science fair in Houston when he was a high school student by solving a really complicated mathematics prize. And this led to an interview on NPR, which was heard around the world apparently because a mathematician in Hungary got in touch with you. So tell us what happened.
Sure. So when I was in high school, I had an early aptitude for mathematics, and it came time for the science fair competitions. And most people were doing wet lab experiments, but that would be, you have to buy stuff, there's like liquids, and there's stuff you have to make. So I thought, why don't I just do a math project instead? It seemed like it may be a bit of a shortcut. And I actually ended up doing quite well. As Julie said, I ended up winning the Houston Science Fair. And then I ended up receiving this letter from this Hungarian mathematician that lived in Texas. And the letter began by saying, "Congratulations on winning the Houston Science Fair. This is quite the accomplishment." Then the next line said, "You should know that the problem that you solved was solved 300 years ago." So I had this incredibly sinking feeling. And then the next sentence read, "But you're not expected to know this because you're living in Beaumont, Texas, and perhaps you haven't had the appropriate type of mentorship. If you want to solve an original problem, please write back." And so the next few days, I just felt really, really, really down. And after 2 days, I just decided, you know what, I'm going to take this person up on his opportunity. And I wrote back. And over the next couple of years, we would write snail mail letters back and forth. And he really instilled in me a love for original research. And he also instilled in me, I think, the importance of taste and aesthetic in science. He used to always talk about an elegant proof versus an inelegant proof. And as you commented earlier, there is something beautiful about science. And I think that was instilled in me at a very, very early age, to try to pursue original research that could also be elegant at the same time.
Well, we're glad that you switched, but fast forward to the time I think you were more or less stranded in your cousin's house. You had no aspirations about mitochondria at that point in time until you noticed the books on the shelf while you were getting ready to sleep, and you picked one up, and lo and behold, there was a picture of a mitochondria in there. So
right. So I transitioned from, I ended up studying math and computer science at Stanford and loved it, and for various reasons got involved in computational analysis of DNA. And so I kind of got hooked on life sciences. And that interest brought me to a specific program at Harvard Medical School that was oriented for students that had math and physics backgrounds. And but it was an abrupt change from Stanford. I'm of Indian ancestry, genetically optimized for the sun. I grew up in Texas. I went to Stanford. And here I was in Boston, and it was cold. There was so much memorization. It was intense, and I wasn't super happy as a first-year medical student. And I remember there's this one brief 5-minute introduction to mitochondria in our pathology class. And the teacher showed an image of mitochondria, which is super beautiful, and commented that it used to be a bacterium, that it has its own DNA, and it just kind of stuck in the back of my mind that this was an interesting topic. But then a few weeks later, my dad's cousin got wind of the fact that I was unhappy, so she invited me to her house near the Somerville Red Line, and as I was walking to her house, the snow started to come down, and I got stranded at my dad's cousin's place on a Friday night. And so she warmed me up, she cooked a nice meal, and then she created a makeshift bed for me. And as I was falling asleep, I saw the Tyler textbook of mitochondria on the bookshelf. And to put myself to sleep, I just started flipping through it. And I remembered hearing about it for 5 minutes in my medical school class. And that night, I would end up reading. I'm not joking. I'm not speaking in hyperbole, but I would literally read 100 pages that night and would check it out for my dad's cousin. And I was like, this is what I want to work on.
So a passion was born. So you have had an amazing series of observations in your lab, and I think we had a wonderful summary of them in our video. But I'd really like to talk a little bit more about why this is important, these signals that these mitochondria are doing. Because when I went through and read your bio and then looked at your papers, I realized that this is a very ubiquitous universe that this signaling may be relevant to. So what's the clinical implication of what you're doing?
Yeah, so I'll just say, I think most of you, when you think about mitochondria, it's called the powerhouse of the cell, right? Because it generates ATP. And people for a long time have linked mitochondrial dysfunction and at the heart of aging, neurodegeneration, cancer, etc. But when people in the last 25 years have closely looked at any particular disease other than primary mitochondrial disease, whereas primary mitochondrial disease is a disease that starts with a problem, a genetic cause in a mitochondrial protein being not properly made. But all these other diseases, people always said, oh, your ATP is not being generated by mitochondria. And that paradigm hasn't really totally panned out. And one of the things that we've recognized is beyond ATP, mitochondria release signals. These things can be signals like H2O2, yes, the bleaching agent, at very small doses, as mitochondrial DNA, lots of metabolites, which I'm very much interested in. And these are just normal signals saying to the rest of the cell, oh, this is how we control health of the cell. These are normal signals to maintain health. And one idea is that in any disease, when someone says mitochondria are dysfunctional, it's not to look at the fact that they don't make ATP. It's these signals either are not being made properly or are chronically activated. Right? And so why that's exciting, and maybe it's a ubiquitous paradigm, as you suggested, is that when somebody says, oh, this disease might be linked to mitochondrial dysfunction, let's think of it as which of these signals didn't get made or is chronically activated, and just fix the signal without worrying about fixing the mitochondria. So if you just take quickly an example like Parkinson's, I was just talking to Dr. Paul about this. So Parkinson's is one of the diseases that's very closely linked to mitochondrial dysfunction. People always say something about ATP, but it could be something else, one of these signals that's either not activated or is chronically activated by the person who's pre-symptomatic that might have mitochondrial dysfunction. It might be difficult to fix that, but maybe if you know the signals, you can intervene at those signals, either turn them on or turn them off. And that, I think, is a new way to think about it. But this paradigm, as you rightfully suggested, could apply to many diseases where someone says, oh, something wrong with mitochondria.
So we're at the 1.0 stage.
We are very much 1.0 stage, and the next generation will be at the 2.0, and it'll hopefully have impact directly in medicine.
So, Vamsi, I have a family member who has a mitochondrial disorder. She was diagnosed as an adult, but she's quite challenged by this disease. And, you know, obviously when we hear genetic diseases and gene therapy, we would like to think, oh, great, CRISPR, let's fix this problem. But that's a lot more challenging when the problem is in the mitochondria. What are the prospects for addressing some of these abnormalities, either in the mitochondrial genes, which, as you said, are just the handful, or the 1,100 genes that are in the nucleus that also interact and have important functions vis-a-vis the mitochondria? Are we on the horizon of therapies that can address some of these issues?
So, I mean, first of all, I'm really sorry to hear that you have a family member with mitochondrial disease. These are super, super challenging diseases. I mean, typically patients will see a median of 7 physicians even before a diagnosis is even ascertained. And we really don't have any truly, truly effective medicines. And as of today, there are 300 monogenic mitochondrial diseases. So it's a very large collection of individually rare diseases. And as you said, some of them are due to mutations in the mitochondrial DNA, and then some of them are due to mutations in the nuclear DNA. Out of those 300 genes, of course, because only 13 proteins are made by the mitochondrial genome, the vast majority of those genes are nuclear. And for those, gene editing, gene replacement, bespoke gene therapies, these are all excellent prospects. For the mitochondrial DNA, CRISPR does not work in the mitochondria. And it's very difficult to get RNA into the mitochondria. And even if you could get the Cas9 protein and the RNA in, mitochondria do not like linearized DNA. It'll get chopped up very quickly, so it's very, very challenging. But one exciting thing is my colleague David Liu at the Broad Institute and Joseph Mojos at the University of Washington in Seattle. Joseph discovered a new deaminase that operates on double-stranded DNA, and David Liu engineered it so that he could fuse it to what are called TALEN, so that they could be programmable base editors. And we help them get it into the mitochondrion so that in a CRISPR-independent way, we can now edit for the first time mitochondrial DNA. I think the good news is we can now model mitochondrial DNA in a dish for the first time. Until now, we had to wait until we saw a patient that has an mtDNA disease and then study those cells. But now, for the first time, we can't make every possible edit, but we can make a lot. And I think that's going to be great for modeling these diseases in a dish or in animals. But I think in terms of using base editing as a therapy, I think we have a solid decade. We're on version 1.0, and we really need to get to about 5.0 before we can really enter into humans.
So we shouldn't be lining up the Bespoke Gene Therapy Consortium to weigh in on this quite yet.
For the nuclear genes, absolutely. For the nuclear genes, but not for the mtDNA.
Got it. So you made reference to your collaborators at the Broad, and both of you were very generous in recognizing your lab teams as really the unit of success here. So while we give the Lurie Prize to individual scientific stars, we all recognize that it's team science. And since we really stand for team science at the FNH, I'd like to just get a little bit more of your perspective on that. When you build your lab, how do you decide who to hire and who to bring into your team? And you're assembling a wise crowd, so to speak, but how do you actually pick and choose? I'm sure you've got people lined up who'd love to work with you.
I mean, I feel super fortunate to be able to lead a super, super diverse, passionate, multidisciplinary team. So I just wake up every morning, lucky I can work with these people. We do have a saying in the lab, whenever we're trying to recruit somebody into the lab, we use a 2x2 grid strategy. Everyone is either brilliant or not brilliant, and everyone is either a jerk or not a jerk. So we try to sample from the upper right quadrant, right? People that are brilliant but not jerks. And people can be brilliant in different ways. They can be really hardworking. They can have a special spark. They can be creative. They can have a high IQ. Many ways of being brilliant. Unfortunately, there's many ways of being a jerk as well. But by sampling from the upper right quadrant, I think we've just built an incredible culture. And I think the other element of our team is that within our group, on the one hand, we have folks that are just pursuing science in a really curiosity-driven manner. On the other hand, we also work on diseases where we have a healthy sense of urgency in the problems that we pursue. So I love the fact that we have both of these elements in a really kind laboratory.
Thank you for that. Now, you and I have talked a little bit about the selection of students and trainees in your lab and mentoring. Share with us a little bit about your philosophy of how you nurture and support your students, your postdocs, and your early career scientists.
Yeah, so I don't have a complicated grid like you do. You're still holding on to your math. I have trouble adding a tip these days. Once they moved it to 20 to 22%, it became tricky. But, for me, it's really simple. I ask a simple question: do you like metabolism? And if you waver, well, I sort of like it. No, I love it. I'm sold, right? Because then they're going to be passionate about what I'm passionate about, which is the big world of metabolism, and mitochondria is at the center of that. And then I really ask them what aspect do they want to study. Some of them want to study clinical diseases. Some of them want to study a very basic question, and I don't hinder them because I figure as long as they like metabolism, we can communicate, and then they can take that in any direction they want, from crazy things like metformin to Parkinson's to cancer, et cetera. I allow them to own their project because I figure if they own their project, they can't blame me when things go bad, right? It's your project. It has to be metabolism.
Now I understand why you wrote a book, Navigating Metabolism. Yes.
It's a great holiday gift. We all got to make a living. Amazon.com. Last year's Lurie Prize winner wrote a scathing remark on it, so there's one person who has a 1 star, but the rest are all 5 stars. Thanks, Andy. Bad joke.
Well, in the course of this conversation, you actually both, in one way or another, really talked about challenging dogma, challenging the dogma that what the mitochondria are there for is to be the powerhouse of the cell, or challenging the dogma about the mitochondria DNA being the important unit of interest, when in fact it's the nuclear chromosome that contains the most of the relevant opportunities for intervention. So talk about the process of science and challenging dogma, and how you face up to the criticism or the status quo that you no doubt have to be a salmon and swim upstream against. I'll start with you on this one.
It's really simple. If your paper gets rejected once, you'll be mad. You'll repackage it, it'll be rejected again, you'll continue to be mad. But the 4th or the 5th time it's being rejected, you should rejoice, because you're onto something that nobody else is onto. And it's true. I can honestly tell you the papers that I cited, and I always cite, that made my career, were perhaps not in those famous boutique journals. One of them, in our cancer, 11 journals rejected it. True story, 11 journals rejected the paper because it challenged the dogma that mitochondria are not dysfunctional in most cancers. And at that time, I'll be honest, I was really depressed and down. It was about 10, 12 years ago. But today, when I get those rejections, I rejoice. Because as long as I'm comfortable in knowing that we did it properly and controlled, because by the time something becomes so big, lots of people are doing it. But if you're way ahead of the game, then most people think you're out to lunch, and that happens a lot to me. Mamsi?
How about your experience in challenging dogma?
So I think there's so many examples of professors that did not get tenure because they had controversial ideas or ideas that didn't fall into the mainstream, and subsequently we then recognized them as being the ones that were really the pioneers. And so there's this old saying that genius is hitting the target that other people can't even see. And I think sometimes when people put up these barriers, you have to just view it as potentially a sign that you're onto something bigger and better than they even recognize. So I think it's a part of our job. We can't take it too personally. Sometimes the papers get in very, very quickly. Sometimes we have to resubmit 3, 4, 10 times. So that's a part of our job.
Part of science, right?
But if I could just say that the point about the tenure is really pertinent because this year's Nobel Prize was given to Carrie Catlin, who didn't get tenure at Penn in the mid to late 90s for the mRNA vaccine, the pioneering work for mRNA vaccines. So that's a great example of someone was way, way ahead of and now is being rightfully celebrated.
Finally honored. Yeah, finally honored.
So.
Well, we're happy to honor both of you tonight with the Lurie Prize. And I hope you feel like these represent rock stars of science and that the work going on in their labs and in their collaborating environments is truly meritorious and deserving of this award. It really represents team science at its best, and I think we're all really proud to have you be part of the FNIH community. So congratulations, and thank you. Thank you. So now, it is my pleasure to invite a wonderful woman to the podium. She's an honorary board member of the FNIH, an honorary awards ceremony chair, and the person whose generous support has facilitated extraordinary scientific progress, not just for her honoring our Lurie Prize winners, but also for the contributions she's made at Northwestern and in various other institutions around the country. So Anne, please join us here. We look forward to your remarks.
Good evening, everyone. What an enlightening conversation that was. Thank you so much, Julie, Nav, and Bamsi. I'm Anne Lurie, and I'm delighted to be here to honor these two groundbreaking scientists with the 2023 Lurie Prize in Biomedical Sciences. I want to digress for just one moment and mention that we have a distinguished group of individuals sitting at our table tonight, and I would like to mention one. We have Eric Nielsen, who is the Dean of the Feinberg Medical School at Northwestern University. Eric, can you just stand up? Okay, doesn't want to stand up, but thank you. My late husband, Bob, and I each felt that helping to empower medical research and medical care was the best way to make a positive impact on the well-being of our fellow men and women. Unfortunately, Bob passed away from cancer at the age of 48, but I know he would have been proud of the charitable work we have supported over the years. 11 years ago, I envisioned bringing greater awareness to the FNIH and to the STEM fields by creating an award to honor a young scientist. And young, I was told, is defined as under 52. The concept was to provide positive models from the fields of science, technology, engineering, and math for young students making their professional life plans. Through my engagement with the FNIH, I had heard stories about the research that scientists such as those we honor tonight were conducting. I made the comment at that point that I wanted to help give them rock star status, because sometimes scientists just go unnoticed. Thus, the Lurie Prize was born. Our jury has identified talented individuals who are conducting research demonstrating groundbreaking achievements in the basic sciences, in basic science. I must admit that when I first heard the term basic science, it didn't sound very sexy. But it's so much more than the name would indicate. It involves figuring out how things work, literally, at the minute cellular level in the human body. Those discoveries will become the building blocks that will solve some of our greatest medical challenges. To meet the men and women who are engaging in this work and are chosen to receive the Lurie Prize is truly a privilege. Let me take a moment to salute our distinguished chair, Dr. Saul Snyder. He's the chair of our jury committee. Saul's done a great job over the years. Any of you familiar with Johns Hopkins probably know the Saul Snyder building. So thank you, Saul, and thanks to all jury members for your unwavering dedication to the Lurie Prize and to excellence in the biomedical sciences. It's a great privilege tonight to recognize the work of Dr. Navdeep Shandell and Dr. Vamsi Mutha. Drs. Shandell and Mutha, I am honored to present to you the 2023 Lurie Prize for your transformational research in mitochondrial science. As we've heard tonight, your independent research has the potential to advance the understanding of characteristics and functions of mitochondria in human physiology and the roles they play in health and disease. I personally had no idea. I remember mitochondria from science many, many years ago, but I certainly did not know they were so important, so thank you very much. And I offer my warmest congratulations to each of you. Please join me at the podium or stand up.
The Foundation for the Institutes of Health presents the 2023 Lurie Prize in Biomedical Science to Navdeep Shandell for his breakthrough contributions to the field of mitochondrial science. Congratulations, Navdeep. The Foundation for the National Institute of Health presents the 2023 Lurie Prize in Biomedical Sciences to Dr. Vamsi Mutha for his breakthrough contributions to the field of mitochondrial science.
Good evening. I'm Steve Paul, Chairman of the FNIH Board, a role that I have treasured and enjoyed for the past 7 years. I want to thank all of you again for being with us this evening, and we have come to the final part of this celebratory event. Now, the Charles A. Sanders MD Partnership Award, named after the FNIH's former chairman, honors his vision to recognize persons and/or organizations that have served as invaluable collaborators and partners with the FNIH. This award, now in its 8th year, is very special as our awardees are nominated by the staff of the FNIH who understand deeply the role that team science plays in changing the face of medicine. Tonight, we honor NIH's Eunice Kennedy Shriver National Institute of Child Health and Human Development, GlaxoSmithKline, and the O'Neill family with this distinctive award. Now, Charlie could not be with us this evening, but he wanted me to read this congratulatory statement to the awardees, and I quote, The 2023 Partnership Award recognizes remarkable partners that have made outstanding contributions to the FNIH's work in support of the mission of the National Institutes of Health. I wish to extend my gratitude to the three awardees, NIH's Eunice Kennedy Shriver National Institute of Child Health and Human Development, GSK, and the O'Neill family for their dedication to scientific collaboration aimed at a common goal, advancing breakthrough biomedical discoveries and improving the quality of people's lives. Thank you. Now, I could not agree with Charlie Moore, and I'm therefore honored to invite the recipients of the 8th Annual Partnership Award to join me here on the stage, and Julie Gerberding as well. Julie. Dr. Diana Bianchi, Director of the NICHD. Brian O'Neill, representing the O'Neill family. And Dame Emma Walmsley, CEO and Board Director of GSK. Dame Walmsley. Now, I think Julie's going to present the awards, and I'm going to read the citations. The Foundation for the National Institutes of Health presents the 2023 Charles A. Sanders, MD Partnership Award to the Eunice Kennedy Shriver National Institute of Child Health and Human Development for their significant contributions to the FNIH's work in support of the mission of the National Institutes of Health. The Foundation for the National Institutes of Health presents the 2023 Charles A. Sanders, MD Partnership Award to the O'Neill family for their significant contributions to the FNIH's work in support of the mission of the National Institutes of Health. And finally, the Foundation for the National Institutes of Health presents the 2023 Charles A. Sanders, MD Partnership Award to GSK for their significant contributions to the FNIH's work in support of the mission of the National Institutes of Health. Now, I'm first going to welcome Diana to the podium and make a few comments. Thank you.
And I will be brief. So thank you so much. It's so appropriate to be representing the Eunice Kennedy Shriver National Institute of Child Health here because we're at the Kennedy Center, and of course, she was President Kennedy's sister. I'm very honored to accept this award on behalf of my institute, and I'd like to thank the selection committee and the board of the FNIH, as well as Dr. Julie Gerberding for her true commitment to maternal and child health research. This award is a testament to team science and the village that it takes to implement our vision. Our staff work tirelessly in pursuit of our mission and vision. Our mission includes not only pediatric research, but also research on reproductive health and pregnancy and intellectual and physical disabilities. Our vision is healthy pregnancies, healthy children, healthy and optimal lives. And we've been very proud to partner with the FNIH on research, as Dr. Gerberding mentioned, using our existing global health network for women and children's health research. The A+ study that she mentioned really took 2 grams of a very inexpensive antibiotic, azithromycin, given to women in labor in 7 different countries, and it halved the incidence of maternal sepsis and death. And if we can do that around the world, hopefully we can also do that in the United States. So the results of the A+ study, which was truly a bridge between NICHD, the FNIH, and the Bill and Melinda Gates Foundation, will change clinical practice and prevent life-threatening infections in postpartum women. We have another study that has started called the Priority Study, which seeks new ways to treat women with postpartum anemia. We're treating them with iron, and postpartum anemia is a major contributor to adverse maternal health outcomes. Lastly, we're looking forward to a brand new endeavor exploring partnerships to develop pediatric medical devices with private and public sector partners. We're very excited to work with the FNIH and our other partners to explore future collaborative opportunities to expand the scope and reach of our research to optimize the lives of children, women, and people with disabilities. Thank you so much.
We will next hear from Brian O'Neill, representing the O'Neill family. Brian?
Good evening. I'm here representing the O'Neill family, so I'm just a spokesperson. I'd like to recognize everyone at Table 12, part of my family who's been a big part of our program. So. We are so deeply honored to be a recipient of the Charles A. Sanders, MD Partnership Award. A heartfelt thank you to everyone at the Foundation for the NIH for this recognition. Our family started our program to honor my dad's brave battle, but also his passion for learning about his disease and sharing his knowledge with the broader kidney cancer community. He was not a scientist, he was a finance executive, but he was also very brilliant. Our fund set out to recruit and support people power because funding basic research is critically important. With FNIH, we found the perfect partner to build a world-class fellowship program 100% laser-focused on research. Progress is also not found in heroic efforts. It's found in sustained commitment and long-term thinking. We are so proud to have sustained our program now for 20 years, supporting 10 research fellows. With our community-based philanthropy approach, we have hosted tens of thousands of people to connect and have some fun. I credit my mom, Suzanne, for teaching our family that hope and encouragement in the service to others is a noble cause. Our fellows also have advanced science in many impressive ways. I'm not even going to mention them all tonight. And many have gone on to seed complementary programs at leading institutes around the world. Matt, you got to help me out here. There's somebody we're not mentioning. Yeah? Okay. This guy's brilliant and not a jerk. He's going to hate this part, but this is where my family is deeply thankful to him for helping my father survive an incredible 7 years with his disease. So he's going to get embarrassed. Rear Admiral Dr. Richard Childs, Assistant Surgeon General, Clinical Director and Acting Scientific Director for the NHLBI. Thank you for shepherding our program. What's amazing is that having an illustrious list of titles has not gone to Rick's head. Recently, he pulled me aside and said, Brian, I'm also just a guy in a lab standing before cells, asking them to love me. We are so grateful for our 20-year partnership with you and your team, and we're encouraged by and proud of all of your work. The force is strong with this one. I often read Rick's research and try to impress him with a smart question. Like, what are you doing to better understand methylation in kidney cancer, and are you learning how to regulate it? Just to be clear, I have really no idea what that means. Methylation, to me, it sounds like cancer and global warming have the same problem. No, but the bigger point here is, are we asking the right questions? There's so much we do not understand about cancer, but better questions can lead us to better questions. We know with certainty that over the next 20 years, working together and taking on bold questions, we will uncover many new and important discoveries with applicability for kidney cancer and beyond. We are emboldened by the mission, and I think that's what it's all about. So thank you to Dr. Childs, our fellows, the team at the NHLBI, and the Foundation for the NIH. We are so proud to be your partner. Thank you.
Okay, finally, Dame Walmsley, I welcome you to the stage.
Hi. Actually, I would like to start by thanking and congratulating Julie for such a magnificent evening this evening.
You know, I think I'm the last speaker, and I'm also, I think, the only one this evening who doesn't call America home. And everyone should know how much the world watches the work of the FNIH. You do truly lead the way. It is an incredible honor to receive this award alongside such prestigious company, and there is no doubt that this evening has been a genuinely inspirational testament not only to what can be achieved in science, but to the FNIH's ability to amplify the impact of scientific endeavor through partnership and through collaboration to the very greatest effect. At GSK, we unite science, technology, and most of all, talent to get ahead of disease together. And the word together is very, very important to us because it takes the best minds to solve the biggest problems. Whether it be scientific advances in malaria, TB, HIV/AIDS, COVID, of course, or the slow pandemic and a big threat of antimicrobial resistance, collaboration has always accelerated progress. And we are constantly facing into new challenges that need new solutions. Today, countries all around the world are facing a demographic time bomb with unhealthily aging populations and an enormous acceleration in chronic disease. And this isn't just a huge health challenge, but a major issue for health systems, for health budgets, for communities, carers, and economies more broadly. But I am a massive optimist, and tonight has been a really good reason to be so, that we are going to find more solutions, bringing our combined talents and ambition across the private, academic, and public sectors to work together to prevent and change the course of disease for patients and alleviate the burden of sickness on society, health systems, and economies. To do this, we need deep expertise in clinical trials, in scale-up, in manufacturing. We need access to data in real time and to patient populations, as well as, of course, regulatory support. And we need pace, and we need access to funding. FNIH has been the catalyst to make these sorts of things happen. And that's why we're all here tonight, to celebrate the progress that has been made in finding solutions to these complex issues and to keep raising our ambitions. At GSK, we are really very, very proud to have supported the work of FNIH over the past 25 years on more than 35 projects. We've been long-standing partners of the Biomarkers Consortium and committed to a number of accelerating medicines partnership programs, including a major research program aimed at advancing Alzheimer's disease treatment. So, Julie, it's quite difficult to look behind me at the same time as speaking to the microphone, but thank you deeply, and to all of the FNIH board, of course, for this award, but most of all, for everything you do and dream of. We look forward to continuing to support the groundbreaking work that gets done here so we can get ahead of disease together for many, many years to come. Thank you very much. You forgot it.
Well, thanks, Dames Walmsley, for that just very inspiring remarks. I want to thank all of the Sanders Prize recipients for their remarks as well. I'm going to turn it over to Julie for some closing remarks.
Well, that was just awesome. Thank you, Emma, Brian, and Diana. Partnership is a privilege, and partnership in the spirit of team science is priceless. We could not do the work that we do in supporting the mission of the FNIH without the caliber, the credibility, and the trust that our partners have in us. So we are deeply grateful for those that we honor tonight, but really we're in a room full of all kinds of partners, and we appreciate your scientific support, your financial support, your wisdom, and all you do to help us be successful. I do want to make one last acknowledgement, perhaps one of the most important ones. We wouldn't have a celebration tonight if it weren't for the incredible leadership of Janelle Lewis and her events team, as well as many other people from FNIH who supported us. So Janelle, I'm sure you're there somewhere. Please wave your hand. Thank you. I also want to give a very special thanks, because I never get a chance to Jane Krumpetich, for whom I've been working since 2010 as my chief of staff. Her experience, her high standards, and her unbelievable patience with me is really the secret to anything that I might accomplish. So Jane, I don't know where you are, but please wave your hand so that we can thank you as well. We've come to the end of our program, so I thank everyone. We're deeply grateful for your support, and together we are building bridges to breakthroughs and better lives for patients everywhere. So thank you, good night, and please have a safe trip home. Thank you.